Get things going. Okay, welcome everybody to this second Idea Community Talk of the Fall 2020 term. Tonight we've got PhD student Miao who works with Kristen Bennett, giving us a very thorough talk and demo on her work on inequity using the ML fairness principles. Just a couple logistical things. We are recording this. This talk will appear later in the week via the IDEA website, idea.rpi.edu. We've got a talk section and a podcast section on the website. Miao you can put questions for Miao in the Slack or questions about the talk, and if there's something that we should call to her attention during the talk portion, we'll do it. But she will be also doing a live demo at the end of her talk, so we can look forward to interacting at that point as well. So without further ado, Miao, take it away.
Hello, good afternoon. I'm Miao, a third-year PhD student with Dr. Bennett. So my research is about how to tackle the health inequities through a combination of the techniques, including machine learning fairness, advanced artificial intelligence, analytics, and optimization approaches. In this presentation, so I will first introduce the problems that I try to solve and why they're important. And then I will talk about some basic formulation about how I derive this equity metrics for the randomized clinical trials. Then actually I will go to give a short introduction on our application called the TrialEquity, and then I will do the TrialEquity demo. After the demo, if the time allows, I will talk about some future work on my research. And then that's a question and answer section, but you can interrupt me anytime you want. Okay, now I will talk about something about health equity first. So the terms equality and equity are often used interchangeably. However, they differ in the important ways. Health equity and health equality do not mean the same thing. Equality means that giving everyone the same thing, but equity means that giving people what they need to reach their best health. So for example, in the picture here, three people of different heights are trying to reach the fruit on the tree. In this case, the fruit symbolizes the good health. The different heights of the people represent the unequal distribution of the social determinants of the health in the society. So if we treat these people equally, we would give everyone we would give everyone the same box to stand on, so only the tallest person could reach the fruit. But if we try to treat them equitably, so we will give them as many boxes as they need to reach the fruit. So in my research, I focus on the health equity instead of the equality. And according to the WHO, the health inequities are defined as the systematic differences in the opportunities that the groups have to achieve optimal health, leading to the unfair and avoidable differences in the health outcomes. Now I will talk about some information about the randomized clinical trial. So the appropriately designed randomized clinical trials can represent the current golden standard primary study design for the determination of the efficacy and safety of the medical interventions, as they are not subject to the same confounding as the non-randomized studies do. The randomization, when performed correctly, guarantees that on average, the treatment groups will be well balanced for both the known and unknown factors, therefore ensuring an unbiased estimate of the treatment effect. The evidence from the randomized clinical trials is used by the healthcare providers to get their clinical decisions and by by payers and policymakers to support their recommendations for the adoption of the new therapies in the clinical practice. In order for the results of such trials to be clinically useful, they must also be relevant to a definable patient population in a specific health setting, a concept that is termed generalizability, which describes the applicability of the study results outside of the trial environment. In recent years, the need to better understand the generalizability of the randomized clinical trial results have been identified across numerous medical areas. However, a comprehensive literature review of the studies that have assessed the representativeness of the clinical trials population has not been undertaken in recent years. The representativeness describes the similarities between the randomized clinical trial samples and the real-world target populations. So in my study, we actually focus on the process from the reference population to the study population. That is the generalizability of the clinical trials. So the causal diagrams have been used extensively to represent the bias in the medical studies. These diagrams represent as directed at acyclic graphs. The absence of an arrow pointing from the variable A to another variable B indicates that the variable A does not have a direct causal effect on B. A key advantage of the causal diagrams is that they provide a mathematically rigorous yet intuitive tool for deducing the statistical independencies implied by the lack of the causal arrow. So has the two models shown here. If we use an example of the randomized clinical trial to evaluate the mental health, then Y here represents the assignment of the subjects into a trial, which targets the patients with mental disorders. T will represent a new drug, Z is the self-reported mental health symptoms, and X is a potential covariate, for example, like the patient age, that affects the assignment of the subject in the trial. The arrow from X to Y represents the tendency of the elder people to seek medical help more often than the younger people. And the arrow from X to Z represents the effect of the age on the self-reported mental health symptoms. In an equitable trial shown on the left, the tendency of the elder people to get help should be controlled when assigning the patients from different age groups into the trial. And in an inequitable randomized clinical trial, however, the covariates influence on the assignment of the participants will make an intention to treat bias for the causal effect of the medical intervention on the response variable. And this implies the significance of the representative randomized clinical trial population to get robust and correct results, and how health equity issues are generated in the inequitable randomized clinical trials. So here, the problems that we try to solve is that how could we help the people in the field of the clinical research to identify the inequitable randomized clinical trials, and how should we quantify and compare these inequities in the clinical trials across different subpopulations, like the different race minorities. And our solution is to design some equity metrics for the randomized clinical trial based on the machine learning fairness research. And there's another research question in the field. So to be able to answer the important questions about whether the treatments or drugs impact some groups differently than others, we must have adequate sample size and representation so that we can make some subgroup analysis. And we really have learned from some important missteps that not all groups respond to the same treatment in the same way. So to design an equitable study, the trial should include a mixture of both those experienced and those not experienced in equity, such that the comparative assessments can be made in the disaggregated analysis or formal subgroup analysis. So we emphasize that all relevant equity factors should be captured at the baseline, which is at the enrollment stage. So design the equity consideration in a mixed population like the figure shown here will include powering the study for the subgroups of interest, especially if these subgroups are included as part of the main study question. So for example, if the researchers are interested in the attributes like the gender, age, and race, it's important for them to recruit enough patients for each of the subgroups defined over these attributes, like in the figure shown here. So if they need a mixture of the population, they actually have to make sure that for each subgroup, like the young people, the elder people, they need also include enough participants so they can get enough evidence for each subgroup. And the characteristics to define these subgroups can be the demographic characteristics like the race and gender, the disease characteristics like whether they have diabetes or hypertension, and the environmental aspects like some risk factors, and also the clinical considerations like they take some specific hypertensive medicine. So the second problem that we try to solve here is how could we help these people in the field to design the equitable randomized clinical trials and fix the inequitable clinical trials. We don't just help them evaluate the inequity in the clinical trials. We want to help them to fix these problems. And our solution is to optimize the equity of the clinical trials to make the equitable recruitment plan based on our proposed metrics and the specific user needs. Yeah, and the third research question in my research is that we want to put the practical tools in the hands of the users, including the clinicians, the clinical researchers, and also the policymakers. We try to target the users in the real world instead of just proposing a solution to the current problem. And by designing a practical tool, it can be more generalizable and scalable for the specific studies of interest. For example, the physicians can compare their clinical studies to a target population in their own community instead of the total US population. Also, some organizations like WHO can compare the clinical trial to the whole world population. So that makes my tool more generalizable and scalable. And finally, is to design a tool with some visualizations is more understandable and efficient for users, such as the physicians, like the figure shows here. Actually, the top equation here, the optimization problem, is solved in my research to optimize equity in the recruitment plan. And the bottom one is the metric derivation for my study. So instead, if I show the physicians these two, they don't understand. But if I show them something like the visualizations here, they can easily detect which group is inequitable or equitable and overrepresented based on these colors. I will talk about the colors later. So our insight here is to view the assignment of a subject to a randomized clinical trial has a random binary classification function. In this way, we can apply all the machine learning fairness metrics to this classification problem and then generate our novel equity metrics for the randomized clinical trial. So, or equity metrics are actually the functions of disease-specific race of both the protected subgroups, such as the non-Hispanic Black people in the randomized clinical trials. And in this function, we need to use the ideal trial race, which is estimated use in our study using the enhance. But it can also use some other sources like the electronic medical records data. And the other data we used is the observed trial rates, which is estimated using the real clinical trial data. So here is an example of the derivation of our equity metric. So compared with a machine learning metric, we or X here, instead of being the selected features of the subjects, it becomes the protected attributes of the patients. And then the true outcome in the machine learning metrics becomes the ideal assignment by a really random clinical trial. And then the predicted outcome from the machine learning becomes the observed assignment in a real clinical study. And then the indicator function becomes the subgroup exclusion and inclusion criteria for the clinical trial. And by mapping all of these factors, then we can derive a metric for the clinical trial from any available machine learning fairness metrics. Like here, we derive the one called the log disparate impact from the disparate impact. To assure that each subgroups can have similar distribution of the protected attributes compared to the target population. And before I do the demo, I will give a short introduction on the TrialEquity application. So our trial application, it helps the researchers, physicians, and policymakers to identify the equity gaps in the clinical trials. And it can optimize the randomized clinical trial recruitment equity through the interior programming, and helps the physicians understand the different patient treatment options. And it can also identify the gaps in the disease guidelines. The inequity evaluation on the randomized clinical trials can answer some questions such as, is the intervention equally safe and effective for all the target populations? And which patient characteristics affect the trial equity? And how much difference does it make in equity? In this visualization, what is called the sunburst here, we categorize the equity levels into 8 different levels, as shown here. So actually for each metric, we have 2 bounds, the upper bound and the lower bound. The upper bound is used to decide the highly inequitable subgroups with the inequitably represented subgroups. And the lower bound is used to decide the equitably represented region of the subgroup and the inequitably represented region. So here we identify 8 inequity levels. So the subgroups may be absent in the target population. This one actually can be fixed if the users allow us to do so. And the subgroup may be absent in both the target and the real clinical trial. And the subgroup may be missing from the clinical trial only. The subgroup may be also highly underrepresented, or equitably represented, overrepresented, and highly overrepresented, depend on their select upper and lower bound for the different metrics. And our optimization strategy on the trial recruitment equity provides answers to the researchers on questions such as how to recruit a representative population into the new trial, how to remedy an inequitable ongoing or conducted clinical trial, The plan details are displayed in a table, and the equity evaluations are additionally provided here as the summarized figures to help the users to identify the improvement of the study equity based on our suggestion. And finally, we allow the physicians to do the patient-specific equity evaluation. It's designed to answer the questions such as, is this trial equitable for my patient? And how similar are my patients to the trial participants? By input the individual patient characteristics into the tool, the system will return a patient-specific equity score and analyze the influence of each attribute on the patient's equity level. So now I will start to demo my application, and I think I see some questions in the chat, but it's blocked.
Yes, Miao. Do you want me to read the questions?
Oh, okay, yeah, thanks.
The first question was on the slide with the fairness metrics. What are the other fairness metrics you can apply? And do you think you will be able to get the same type of analysis?
So you mean what different kind of the equity metrics?
Yes, what type of different equity metrics can you apply? Can you give some examples?
Yeah, I think actually we also have examples in the application called the adjusted equal opportunity and the quality indicator, I think is called. Yeah. So
will you change the sunburst based on the metrics?
Yeah, the sunburst is based on the metrics. Yeah, it will change. Yeah. In the application, actually, I can show you how it changed. Yeah.
And then my next question was I think with like the sunburst slide, how are the bounds calculated for each equity loop?
Sorry, I didn't hear. What?
How are the bounds calculated for each equity group? Yeah. And is it based on a study?
Okay, actually, for the log disparate impact, the bounds are decided from the literature. So usually people use the 80% rule. So that is 0.2. And then we do a relaxation. So the higher bound becomes 0.4. I think this one is also from the literature. Yeah, but actually it can depend on the user. You can adjust it based on your situation.
Okay, and then like I also want will they be same for 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 multiple studies? Will there be a skew in some studies maybe? Maybe some studies are skewed for like a particular reason, right?
You mean so you mean some study try to include the minorities more?
Yes, exactly.
Oh, okay. So currently our metrics just show you the different inequity levels here. We didn't consider about the inclusion exclusion criteria such as they intentionally to recruit more non-Hispanic Blacks or Hispanic people. Yeah, but
I think it would be a simple sort of a conditioning that you need to introduce into your model. You need to be able to condition it based on the inclusion exclusion criteria.
Yeah, actually that's our next step.
Okay.
We currently just show you the equity measures.
Okay, sounds good, Miao. Thank you.
Yep. Any other questions?
Why don't you keep going, Miao?
Okay. So where is my? Sorry, it takes some time. So currently this application is only available from the IDEO server. So we need to use VPN if you want to play with this one. Okay, now this one is my application called the TrialEquity. Yeah, so this one, this application is actually developed for two different types of the users. The first one is the researcher, and the other one is the physician, because in the real world, actually these two types of users focus on different tasks. So we can start from the researcher part. Yeah, so to start analysis, on this page, you have two tasks to do. The first one is you need to decide the disease that you want to explore. So actually, if you don't know what you should input here, you can click this info icon here to get more information. Like what shows here, it tells you that a selection of multiple diseases is allowed. So this means that actually you can explore the clinical trials that focus on the population of more than one disease, like the type 2 diabetes and hypertension together. And then after you select the disease that you want to explore, here you need to upload the target population information, which we need to use to estimate your target population statistics. So here, when you click upload your own target population data set here, you can just upload whatever you have and you want to use as your target population estimation. But also you can select to use or sample data, that is the enhanced data. So for the enhanced data here, so we actually have, we upload multiple files, and you can also upload more than one if you choose to select your own. Here, the three files means the first one is actually to estimate for the patient characteristics that not include any laboratory results. So this one, you can rename all the files to better understand. So for example, this one I can just remind me has the non-laboratory factors. And the second one is for you to estimate the population data on using only the lab results factors. So this one is called the lab. And the final one is if you contain both category of the factors. So I can see all, and then it will remember your renamed files. And then on this page is very similar to the previous one. This one, you need to upload your randomized clinical trial data. So you can upload your own, or you can use or sample data to explore the tool. So here, we have multiple files, so we can upload two different files here, and one is called the spring and the other one is called the OHAT, because I already select the disease type as the hypertension. So so it returns me these two files are all for the hypertension. And then we can go to the main page for the for this application here. So before you start analysis, you also need to select which file you will need to estimate your target population data. And then you need to select the current file that you want to analyze. And I select the spring here, and then you need to select which task you want to perform. You have three different tasks, has the user, has the researcher. And it explains here, for example, for this first task is equity evaluation, and it measures the equity level of the different randomized clinical trial subgroups with selected patient characteristics. And then I close this one. And now you need to choose the patient characteristics that you are interested. So for example, I may interested in all the demographic features. So I select gender, age, race, and education. So it will show you a sunburst here for you to explore the equity levels. So we actually allow you to change the different metrics here, and also change the different the thresholds for the metric. And also, if you think this figure is a little bit small, you can zoom in to see the details more clear. And for the sunburst here, so there are some instructions here. So when you're hovering the mouse, you can see more details about each subgroup, including their equity level, the ideal rate of this subgroup, observed rate, and the exact number of the participants in this clinical trial. And if you click this subgroup, you will get a mass figure based on this specific subgroup, and also based on the metric that you select. So on this figure, you can see how far your randomized clinical trial subgroup is from the equitable region. That's the teal region here. So you can see here. So it's maybe kind of close to the underrepresented region, but it's still far from the equitably represented region. And also, so for each subgroup, when you click them, I will return your report here. So for example, I click here. So the report will dynamically change for you. It will tell you what study, that's the spring, and then you have your subgroup is female, age over 59, and it tells you the equity value from the metric log disparate impact is about -0.02, which means that this subgroup is actually equitably represented. And the ideal rate of the group is 0.291, and the observed rate is 0.287. So in a random clinical trial like the SPRINT, which has the 9361 participants targeting the population of the hypertension, so we expect this subgroup to have around 2,725 people, but your trials contains a subgroup of 2,684, so which is not significantly different from the target population rate, so it's equitably represented. Yeah, and we have the reference of the different colors, so you can reference what these colors mean whenever you perform the tasks. And we can go to the second one. For the second one, we try to do the comparison between the different studies. So here we selected 2 studies, and you can also do one, whatever, how many multiple files you have. So for example, for here, it's very clear that if the researcher only care about the gender, by comparing the univariate on the gender of these 2 studies, we can see that OHAT is much better because the female and males are equitably represented, but in the SPRINT, the female is underrepresented. And if we go to the age for another example, so this actually very common in this hypertensive clinical trials, because researchers try to include more elder people than the younger one, because they assume that the older people has higher rate to get the hypertension. So in both of the case, they include too much people over 59 regarding to our metric, and actually both the younger and the middle-aged people are highly underrepresented, and the younger one actually absent from the study. And we also do other things like the race, ethnicity, and what's interesting here is based on our metrics, some minorities like the non-Hispanic Black people are highly overrepresented in the study, and the non-Hispanic white, which are traditionally advantaged groups, are actually underrepresented and highly underrepresented in the OHIDE study. But other minorities, maybe like the non-Hispanic Asian and other racist groups, are still underrepresented. And we also do the clinical characteristics, which are not always examined in many studies. They only care about the demographic ones, and we also do the clinical characteristics. So, and actually, Oh, okay. So for this one, because we already know that these two studies about hypertension. So when they do the recruitment, for example, for the sprint, they actually make it equitable regarding to the total cholesterol, which is related to the hypertension. But actually in the summary, when you see the multivariate analysis, when you add other patient characteristics additional to the total cholesterol, it becomes inequitable for some groups. So usually the studies only care about one level characteristics. Yeah, and now the third one is to help the researchers to improve their equity in the study plan and also provide them remedial additional recruitment plan. So here, for example, I want a new study about all the demographic features, and let me do a simple one, just maybe gender and race, and on the southern people, then you do the calculation. So here, like what I showed in the slides of the mixed population part, I provide you about how many people in each subgroups, like the female Hispanic, and also the higher level, like the female level. So we try to make sure that the females from different races can add up together the same as this female level. So this is one constraint of our optimization problem. And then if you don't want to look at the numbers here, I will also show you the numbers figure here. So when you see at this, it's very clear that all the subgroups are equitably represented. And if you want to make sure that, yeah, actually for this one, it's hard to explain, but yeah, maybe I can. Yeah, so for example, if we have three different protected attributes, so this level means the female group, and this one means the non-Hispanic Black female group, and this one means the non-Hispanic Black females over aged between 40 to 59. Now you want to know how about if I consider about the race or ethnicity only, then you can rearrange this actually. You can rearrange like the race or ethnicity, then this one represents for the race only, and then they go to another level to adding in the gender, and then at the age you can rearrange these orders to generate different results. That's for the new study, but how about if my study is already shown to be inequitable? Then here, yeah, and there is a limitation for my additional recruitment. I can only recruit about 1,000 people, maybe because of the budget or something. Then I can also provide you a remedial replan on the additional recruitment. So I show you how many you have before and how many you should recruit based on your 1,000 limit. And that's the total people in your new study if you follow our plan. And I also show you the sunburst too here. So it's very clear that, for example, for the non-Hispanic white, by following our plan, it becomes more equitable here. And some other groups like these subgroups here becomes equitable. And if you don't have the limit, you can just increase the number to make the study becomes more and more equitable. Yeah. And then, so that's the recruitment plan. And then we can go to another task. Yeah. So if you're a physician, all these other things are the same. So the first task for the physicians are to use some simple figure like the bar charts to show them the comparison between the target population and the study population. So by looking at this, actually I can change to another one for better demonstration. So here you can see very clear that if you do that the young people here are highly underrepresented by just looking at this very simple figure here. And when you see this group here, you may think, oh, maybe it's equitable. But if you want to make a further decision, you may need to go toward equity evaluation to to do a deeper analysis. This one is exactly the same as the one for the researchers, so I will skip this one here. And the last task for the physician is to do the patient-specific equity analysis. So here you can, there's also based the demographic. So if my patient is a female aged over 64 and is from Hispanic group and also has some high school graduate education level, then I can calculate the patient equity for this individual patient. Now you can see very clear that this patient is actually fall into the highly underrepresented subgroup. So based on this work, the physician may think that maybe this study I used here called ACCORD for the type 2 diabetes, maybe not very suitable for for this patient, this individual patient. Then we also provide you with how each different factors from this individual patient influence her equity score. So from here, you can see that the education level for this patient is actually overrepresented. So, but for the other three factors here, like the race, the gender, and the age, put her into the underrepresented subgroups, and especially for the race one, and is highly underrepresented. So based on this analysis, it seems like this intervention may not be applicable for this individual patient since there is no enough evidence from the current study. But sometimes, for example, like if I change my patient to the male and over 64 and non-Hispanic white. So here you can see that actually for this patient, this study is equitably represented the people similar to him. And look at here, most factors are overrepresented for this group. So if the physician think the overrepresented is not influential for his patients, this study may be useful for this patient, but our goal here is to show some equity results for the physicians, but to make more decisions, they need to use their specific domain knowledge. So we don't try to help them make decisions. We just provide some guidance on the equity situation for their patients. And those are all the functions of my application. Do you have any questions?
I'd just like to say, Miao, you did a really fantastic job going through the app and explaining all the features and the significance of all the features and giving lots of great examples as you were doing it. Nicely done. And it's a challenge for such a complicated app. There's a lot here. Does anybody have any questions for Miao?
I just wanted to say, I really like that gauge chart. I looked up, what it's called, because, the one that you had for the equity score, that's very nicely done. I guess it's very easy to understand or convey the main point of the app. So well done.
Is that the speed limit one?
Yeah.
Yeah,
it's like a speedometer, but apparently the technical name is gauge chart.
Okay.
Correct me if I'm wrong, Miao. I guess you know more than I do about these charts.
Yeah, it is called a gauge.
Yeah.
Although I do have one criticism, yeah.
Oh.
You did not emphasize the fact that you did this in R.
Oh yeah, that's our shiny. Yeah, that's right.
Miao, how did you choose these charts? How did you decide what charts to use?
How did I choose this one?
Yes. How did you choose the type of charts? Was it all Kristen, John, Dan, everyone getting together and deciding?
Usually, I just do some literature review and look at the function of different charts, and then I try to code this one in the app, and then I need to get the suggestions from Dr. Bennett or Emma or other
nice.
on the design. Yeah.
Actually, why don't you? So, Shruti, that's a great question, and one in particular that might merit discussion is the sunburst. Because that's a kind of a reoccurring theme is the sunburst, and I think during your presentation part you made a great point. I think it was early on when you talk about you've got those equations, all the probability equations and all that kind of stuff, and you contrast that with the information that's presented in the starburst or sunburst. Is it sunburst, starburst?
Yeah, some I think both of them.
Yeah, and I think that was a great that was very early on. Also, I think in your app development, right? When you were still kind of using notebooks and that sort of thing.
I got a question. Miao, when I was listening to your talk, which I thought was great, I was wondering, like, if it was inequitable and you have the original clinical trial data, could you make an estimate that was specific for that population? Like a stratified estimate of the treatment effect?
So what do you mean? I have the original study?
Well, you have the original BioLink data.
Yeah,
so
I have.
You could potentially estimate the treatment effect.
Oh, you want me
have to be careful, but
you want me to estimate the treatment effect?
Well, I mean, we're just measuring equity, but I mean, or I guess would it make sense to estimate the treatment effect? Could you fix the study, is what I'm saying, for that subgroup if there was enough data?
Yeah, I think maybe we can. So currently we only focus on the response independent participate data. So that is not related to the treatment effect.
I'll have to think about that. I mean, I'm not saying doing it. I'm just wondering, like, we could maybe do that, but you have to be really careful because you'd have to worry about the power and all that kind of stuff.
Oh, yeah. That's right, yeah. Yeah, and I think we can do it, actually.
Well, I just wonder if that's a reasonable idea, like, well, it's inequitable, but hey, because they might deliberately oversample some populations to make sure that they're.
Yeah, but you can also do the meta-analysis from different studies, maybe.
Yeah, something like that. Yeah, we'll have to think about that.
Mm-hmm.
Right now, we just measure. We don't worry about fixing, except for the treatment.
Yeah, the recruitment plan, yeah.
Mm-hmm.
I have another question, Miao, regarding maybe the comparison. How do you compare the study data to that of the patient? Do you do a sort of, let's say, do you do a sort of like, do you do like a sort of roll up of the data?
You mean comparison for?
How do you compare the equities? Do you compare it at the individual levels, or do you compare it at I don't know how to put this, or do you compare it at a aggregate level? Yeah, aggregate is the word. Yeah.
compare the equity levels? So you get from different studies or for different subgroups in the same study?
So basically, I'm talking about study to patients, enhance to studies, patients from the studies, patients from enhance. Do you take a sort of like a aggregate to compute the equity, or do you compute it at like basically individual level? I might not be understanding the whole framework, so
Okay, yeah. So actually, for the enhance is used to estimate the target population data. So it's different from the randomized clinical trial.
Yeah.
And for the study population, we need to use the randomized clinical trial data to estimate the study population.
And do you use individual level patient data, or do you use aggregates?
For the BioLink, I think we have the individual patient data, but actually we, to perform analysis using our metrics, we only need to have some statistics, like how many % for each subgroup. Yeah.
Okay, that sounds good. Thank you.
Okay, so if there aren't any additional questions, I think we should thank Miao. Thank you very much for a very in-depth talk and demonstration today. And we look forward to a talk at this time next week. Joe, I think you're talking next week, aren't you?
Yes, I'll be giving a talk on privacy attacks and related to differential privacy.
It's going to be awesome. And look for an announcement Friday or over the weekend of that talk. All right. Thank you, everybody. Have a great evening. And thanks again to Miao.